All right, I think it's time that we get started now that we have most of our audience members and the speaker on call. Okay, so good morning, everybody it is 18th November here in the United States, and we will be beginning our very first collaborative event with SOHO Italy. And we are so fortunate to have the president from SOHO Italy here, Claudio, as well as the brilliant speaker, Dr. Angelo. So just to give a brief outline of what the event entitles today. So today I would be talking a little bit about SNEV and give the welcome remarks to begin this event. And then Claudio would be discussing about giving us an introduction about SOHO Italy. Just a quick housekeeping note, I would request everybody to please mute themselves. That would be really appreciated. So Claudio would be also discussing about melanoma and the workshop they had in July 2021. And then I would be introducing the speaker, and then Dr. Faini would be giving his talk, and then in the end, we would be having our Q&A session. So for the Q&A session, I would request everybody to post their questions during the talk in the chat box. And in case if you have any further questions, you can even unmute yourself during the Q&A session or raise your hand so we could unmute you, and then you can ask your question directly to the speaker. So, Vera, I would like you to mute yourself, please. Thank you. Yeah. So just a brief introduction about what SNEV is and how did we begin. So SNEV is basically a virtual network, which has been running by early career researchers with a main goal to connect EV students and early career researchers from all around the globe, multiple universities, research institutions. And the major initiative of SNEV started in June of 2020, where we saw that there are so many other organizations such as ISEF, Train EV, Web EV Talk, which tries to connect early career researchers, which are led and run by professors. So we thought, how about we come up with a platform that connects EV students worldwide, especially during the pandemic when everything was slow and everybody wanted to be more connected to science. So a little bit about the history of SNEV. We started our organization in the 2020 of June with a mission to connect students from all around the world. And our goal has been to basically have a platform so that students can discuss and network and collaborate with each other, as well as early career researchers, to basically learn more about the EV field and what are the current loopholes or gap of knowledge that they can address in their research or PhD projects or even undergraduate projects. So of course, our team is a big team comprising of brilliant people all across the globe. It's not just one person's effort, it's a team effort. It takes a village to acquire what we have acquired today. So here we have listed the chief executive committee members of the year 2020 to 2022. And we also started having a group of postdocs who are helping us in a lot of ways. So that's all about our team. And we are very grateful for all of you to join us today. And on behalf of each and every chief executive member of our team, we welcome you for today's event. And I would now like to pass on to Claudio for discussing about SOHO Italy and giving us an update, giving us an introduction about the organization. Claudio, you can
Thank
take
you. Thank you. Many thanks. It's a great pleasure to be here with you for this great event, and in order to consolidate the collaboration which we are which we have starting through our event, and I hope and think that will go forward also in the next future. So I hope that you see my slide.
Yes, perfect.
Perfect. I would like to introduce what is SOHOItaly. I am Claudio Cerchione. I am a physician, also PhD, so researcher, and I work currently in North Italy in a research academic institution called Derived, which is located in Meldola, which is a little town close to Bologna in Emilia-Romagna. And what is SOHOItaly? It is a new scientific international society which aims to integrate the clinical scientific activity of different countries in order to improve the clinical approach, clinical and preclinical approach, I would add, in our patients affected by hematological malignancies. Our dream, our end point of this association is that our patient will not need to travel anymore to receive the best and the most effective and the most personalized treatment. This society has started these activities in September 2019 when we organized the first international conference located in Italy. And the society was created together with the leukemia department of MD Anderson Cancer Center, that you know very well is one of the greatest institutions in cancer field in all the world. And we had the honor to create this society together with Agop Kantarjan, which is the lead of leukemia department, is our honorary president, together with me and Giovanni Martinelli. This is the scientific board you can see in this photo that we took in Rome at this first event, in which you can see all the leukemia department faculty members, in particular from left, Guillermo Garcia Manero, Naval Daver, Tapan Kadia, Farad Ravandi, Marina Konopleva, Courtney Dinardo, Elias Jabur, Alessandra Ferraioli, Kantarjan and his wife, and me and Giovanni Martinelli. We have the patronage of many scientific and no-profit society in Italy, so also patient association. We are promoting our activity, and we love to introduce also our patients and caregivers in our educational events, and that's why we try to share also through their association and their family members. It is organized together with my institution, which is Istituto Scientifico Romagnolo per lo Studio e la Cura dei Tumori, which is the Cancer Institute of Romagna, so the region in which it was established in 2007. So it is a young institution. Was a scientific director is Giovanni Martinelli. IRST aims to provide in Italy the state of the art in diagnosis and treatment of hematological malignancies. It is a public institution, so offering all these tools for free for all patients with the collaboration of Italian public health system, and providing also a part of a banking foundation who helps us to promote our projects. It's a multi-speciality center, not only hematology but also oncology, with the semi-complexity clinical specialties such as radiometabolic therapy, last generation radiotherapy treatment, new cellular therapies, immunotherapies, and so on. And it is inserted in the Romagna Oncology Network, in Comprehensive Cancer Network of Romagna, promoting clinical trials and research, but also treatment outside from clinical trial. With SOHOItaly, we have an official journal, which is Hematology Reports, in which I also want to strongly encourage you to submit your works and also to propose as a collaboration. It is indexed journal, and soon we'll receive also an impact factor. Giovanni Martinelli is the editor-in-chief, and we publish for every event of SOHOItaly our abstract book, educational book with educational papers, and we are also promoting How I Manage educational book written by the leader member of our faculty. And we aim also to publish there the conference and workshops reports, such as we could publish something about our events together with the SNEV. So it's promoting also the schools of hematological malignancies, event dedicated to different diseases. We started with acute leukemias, multiple myeloma, CAR-T, but also this event about extracellular vesicle and the role in patient with multiple myeloma is a focused event. Our aim is to create an international team, integrated the board composed by international event with the group of young researchers. In particular, during the SO schools, we select a cantera, under 40 cantera of several neoplasm, which is selected through CV submission and evaluation and their projects in order to promote the activities of our young investigators, and we give prizes for young investigators, which are our guests for the organization and for the participation to event. Every event are free for young, and moreover, we also want as guests in our live event our young investigator. We have some social network. In particular, we decided to organize this event about extracellular vesicle in multiple myeloma, connecting the basic research and the clinical research, connecting also two different worlds, Italy and Australia, where they have a really strong clinical and preclinical group about this topic, led by Professor Spencer, and we connected our institution and the institution of Bari, led by Professor Vac, and Antonia Reale, which is Italian, but now is working in the group of Professor Spencer, was our connection, the hybrid connection between these two worlds. And we tried to integrate in our event all the figures which are working currently in this approach and in this research. Antonia is here with us. Annie? No, so I can continue also presenting a little bit what we did. This was the faculty as I introduced it to you, and this was the program of the event, in which we had a plenary session in which we presented all the clinical and preclinical part about the management of multiple myeloma, the current hot topic in research and in clinical base, the unmet clinical need, and then we focused about the role of extracellular vesicles in multiple myeloma. But as in So Spirit, we selected oral communication from the contents that we received by our young investigators, and that's why we had 4 selected oral communications. We had a vote from the followers, the subscriptioners of SOHOItaly, and Dr. Angelo Faini won the event. He was the winner, and his work was about the modulation of positive and negative effectors induced by soluble and MB-bound therapeutic anti-CD38 antibodies in multiple myeloma. And as we agreed together with your group, we decided to promote his wonderful research in a dedicated event, which is today. And I want to thank all who promote our activities and who help us to realize them. So all the society that you see, also Italian Society of Hematology, Italian Society of Experimental Hematology, and the SO USA, because we are a small part of SO from United States, which is located in Houston in Texas. And the organizing secretariat, which is ProEventi, which help us to integrate in our activities. So thank you for this presentation in order that you know what is this entity, what is SOHOItaly, and I am open at the end for questions also about our society and about I hope that you will subscribe to our society to follow our clinical and preclinical activities. Thank you, Kshipra.
Awesome. Thank you so much, Claudio, for the fantastic introduction about SOHOItaly, and we're so excited to have this collaborative event, especially to honor Dr. Angelo's incredible work and the winner he was in your conference. So now, quickly doing a brief introduction about Dr. Angelo. So Dr. Angelo graduated with honors in medicine and surgery from the University of Turin. He attended the MD PhD program and developed his research in the laboratory of immunogenetics at University of Turin. And Dr. Faini's research focused on the mechanisms of action of therapeutic antibodies in multiple melanoma. He has collaborated with the Dana-Farber Cancer Institute at Boston and was also a visiting student at Howard University. He has published seminal papers in both international journals as well as conferences, and he has presented his work both in American and European conferences. And he's currently a resident in the medical genetics department at the University of Turin. And we're so fortunate to have Dr. Angelo with us today for his presentation. And you can take it away, Dr. Angelo.
Thank you. Thank you very much, Kshipra. I'll try to share my screen now, if I can. So, can you see it? Can you see it?
Perfect.
Yes, perfect.
Okay, so once again, thank you for the introduction, and good evening to everyone. Today, I would like to talk to you about my experience with microvesicles in the multiple myeloma disease model. And in particular, I will discuss how the treatment with anti-CD38 therapeutic antibodies can unveil a possible role of microvesicles as mediators of communication between the tumor and the immune system. Before entering the main topic of this talk, I would like to give some background about multiple myeloma, so as to allow also those of you who are not so familiar with this malignancy to fully understand what's coming next. Multiple myeloma is a malignant proliferation of plasma cells, and these cells produce large amounts of monoclonal immunoglobulins, which all share the same isotype and specificity. These immunoglobulins, also called M protein, accumulate in the organism, thus damaging the tissues and impairing organ function. Also, this uncontrolled proliferation depletes the cell populations in the bone marrow, enhances bone reabsorption, and reduces hematopoiesis and immune activation against pathogens. This observation can explain the most common signs and symptoms associated with multiple myeloma, such as anemia, impaired renal function, pathological fractures, hypercalcemia, and many others. Multiple myeloma is considered a disease of the elder people, being its median age at onset around 70. However, more and more diagnoses in young patients are being reported, and also possible correlation with environmental factors has been proposed, also considering the fact that the incidence in industrialized countries is much higher than the average. In the majority of cases, multiple myeloma is not an aggressive and rapidly evolving disease. It is rather silent and slowly progressive until diagnosis. However, as of now, no curative options are available, and patients undergo sequential treatments, which only last until the disease is no more sensitive to the compound used. For this reason, a deeper understanding of its pathology and of the mechanisms of action of the available drugs are much needed to improve patient outcome and survival. A characteristic feature of multiple myeloma cells is their surprisingly high expression levels of one molecule, CD38. CD38 is expressed at various levels by many cell types, such as T and B lymphocytes, macrophages, NK cells, and many others. CD38 is a surface molecule, almost ubiquitously expressed, and weighs around 45 kilodalton, and exerts multiple functions. It's primarily an ectoenzyme, which metabolizes NAD to cyclic IDP ribose and IDP ribose. And the latter is then metabolized by CD203 and the product by CD73 into adenosine, which is an important tolerogenic factor. CD38 is therefore part of an alternative pathway of extracellular adenosine production, flanking the canonical one, which instead exploits the enzymatic activity of CD39. CD38 is also a receptor capable of modulating molecular pathways such as those of BCR, MAP kinase, and Toll-like receptor. Finally, CD38 is a heterotypic adhesion molecule that regulates the transendothelial migration of immune cells. The interest for these molecules comes from the fact that since multiple myeloma cells score the highest expression levels, CD38 was chosen and adopted as a target for monoclonal therapeutic antibodies, which represent nowadays a cutting-edge therapeutic option for multiple myeloma patients. The first antibody to be approved was daratumumab, a human IgG monoclonal antibody that is now used in clinics both as a monotherapy and more often in combination with other drugs. Daratumumab acts on myeloma with different mechanisms of action, such as complement-dependent cytotoxicity, antibody-dependent cell cytotoxicity, antibody-dependent cell phagocytosis, immune modulation, and the induction of apoptosis. However, daratumumab may sometimes be ineffective and resistance eventually occurs. This was the starting point of our work, whose aim is to better understand the interactions taking place between the antibody, multiple myeloma cells, and the immune system. This will hopefully allow us to expand the possible uses in therapeutic combinations of the antibody. The first observation that we made is that daratumumab induces, upon ligation to its target CD38 on the cell surface of myeloma cells, a redistribution of the target molecule, which coalesces into polar aggregates. Morphological changes follow, which lead to the production and release of antibody-covered microvesicles. In the top part of the slide, you can see confocal pictures of multiple myeloma cells exposed to daratumumab. On the left, cells were kept at 5 degrees Celsius, that is in a condition of membrane stability, and a uniform fluorescence was observed. CD38 is in fact uniformly distributed on the cell membrane. The two figures on the right are instead cells that were kept at 37 degrees Celsius. In this case, a capping was observed and molecules on the MVs forming on the cell membrane were observed. Below the confocal pictures, you can see a schematic view of the events I have described to you. The microvesicles released from multiple myeloma cells were then analyzed to evaluate their size and concentration. The nanocyte analysis showed that the microvesicles have a rather uniform dimensions of about 150 to 200 nanometers, and that the treatment with daratumumab does not substantially modify it. Daratumumab, on the other hand, increases their concentration approximately 4 times. We then evaluated their phenotype, and the microvesicles were analyzed with an appropriately set cytofluorimeter, and the results showed that there is a coalescence and therefore an enrichment of some molecules anchored to the same lipid rafts. Among these, in addition to CD38, which is the target of the antibody, there are CD39 and CD73, both involved in the pathways of adenosine production. However, other molecules were observed on their surface, such as CD55, CD59, and PD-L1, which belong to the families of complement inhibitory receptors and to immune checkpoints. We repeated these experiments using microvesicles derived from bone marrow samples of myeloma patients treated with daratumumab and daratumumab plus retinoids. The results show a similar increase in the expression of the molecules, confirming that what was seen in vitro also happens in vivo. The main changes are highlighted at the end of the treatment with daratumumab, while the addition of retinoids does not seem to change the expression profiles. The next step, before focusing our attention on the content of microvesicles, was to assess what effect the daratumumab treatment, and therefore also microvesicle release, had on myeloma cells. Using the RNA sequencing technique, we observed an upmodulation of genes related to metabolic processes of carboxylic acid, small molecules, and many others. On the other hand, the main down-modulated genes belong to the pathways of cell cycle and mitosis. This data can be explained on one side as a cellular response to the energetically expensive release of microvesicles, and on the other as an anti-proliferative action of the antibody. We then focused our attention on the content of microvesicles. We performed the same type of analysis, looking for any differences in the microRNA content of microvesicles. As shown by the heat map on the left and by the principal component analysis on the right, daratumumab causes a differential distribution of the microRNA inside the microvesicles. However, the interpretation of these data was particularly difficult, and assess what the differences observed meant was a really hard task. So to bypass this issue, we asked ourselves what was the biological significance and the fate of these microvesicles once they were released from the myeloma cells. So we hypothesized that these microvesicles can diffuse through the organism and body fluids, interacting with other cell populations, and in particular with the immune system. To verify this hypothesis, we performed internalization flow cytometric assays, and myeloma cells were stained with the vital DID dye, which is a membrane intercalator and uniformly stains all cell membranes. The cells were then treated with daratumumab or control IgG, and microvesicles were isolated and incubated together with different subpopulations of the immune system. The results indicate that macrophages have the highest internalization efficiency, but they're not selective, while NK cells and myeloid-derived suppressor cells have an intermediate internalization efficiency, about 40 to 50%, but preferably internalize antibody-covered microvesicles. We therefore focused our attention on these two cell subsets. We then confirmed what we saw from a quantitative point of view, also qualitatively. The confocal microscopy pictures in the slide represent NK cells or myeloid-derived suppressor cells stained with cell-specific antibodies. The red dots inside the cell are microvesicles derived from stained multiple myeloma cells, and this confirms their internalization. The nuclei are shown in blue. Once internalization was demonstrated, the next step was the molecular analysis of the effects it induced. The focus was primarily on NK cells, as they are the main positive effector against myeloma and often paradoxically reduced in number after daratumumab treatment. RNA sequencing showed an upmodulation of genes involved in the immune response, in the regulation of apoptosis, in chemotaxis, and inflammatory response. Among these, there are numerous chemokines, BCL2, STAT1, NF-κB, TNF, IL-8. The down-modulated genes instead belong to gene families responsible for mitosis, cell cycle, and nuclear division, such as cyclin A2 and B2, Aurora kinase, IL-12B. This data seem to indicate that NK cells, in response to daratumumab, although less proliferating, are more activated, reflecting the efficacy of daratumumab on one side and the paradoxical decrease in the number of NKs observed in vivo on the other. We also consolidated these observations with functional experiments with soluble daratumumab and microvesicles obtained from cell lines. In this slide, the first row of graphs summarizes the results of the experiments performed with soluble daratumumab, while the second row summarizes the data obtained with the microvesicle-bound antibody. The results show an increase in the expression of the early activation marker CD69 and the degranulation marker CD107, while the production of interferon gamma is reduced. We also wondered if the observed effects could in some way be due to the binding of daratumumab to the Fc receptor present on NK cells. To this end, we produced the FAB2 form of daratumumab and repeated the experiments. As you can see, this last formulation of the antibody seems to have no particular effect, which led us to think that it is the combined action of the binding with the Fc receptor and with the variable region that induces synergically the observed effects. These same results were obtained using microvesicles derived from bone marrow samples of patients treated with daratumumab and again with daratumumab plus retinoids. Also in this case, what previously observed with cell line was confirmed. And once again, adding the retinoids to therapy does not seem to change significantly the results. Further validation was carried out by analyzing the proliferation of NK cells exposed to daratumumab. Also in this case, the results of functional experiments confirmed those of molecular biology, highlighting a reduction in proliferation in response to daratumumab. This decrease was observed, although to a lesser extent, even with the control human IgG, highlighting the important role of the Fc in NK response to antibody. Moreover, these data are relevant per se, given the high amounts of antibodies administered in clinics, which can have substantial impact on NK proliferation. Trying to complete the picture, we then turned our attention to the main compartment of negative effectors in the bone marrow niche, that is myeloid-derived suppressor cells. What we know from literature is that daratumumab seems to induce a decrease in their number and in their activation, thus reducing immune suppression. However, our results seems to go against the tide, and as you can see on the left, daratumumab does not affect so much the viability of myeloid-derived suppressor cells, either in its whole form or in its FAB2 form. Furthermore, we observed how daratumumab induces an increase in the expression of iNOS and in the production of nitrates. And as shown in the scheme on the right, both are markers of myeloid-derived suppressor cell activation, thus suggesting a possible and paradoxical pro-tumor effect of the antibody. However, the literature about this topic is still conflicting, and of course, further investigation is needed to understand the biological meaning of what we have observed. And this is of high importance since myeloid-derived suppressor cells represent a high percentage of the cells in the bone marrow niche of myeloma patients. And studying their dynamics in the presence of the antibody can be extremely useful to understand what the mechanisms of non-response or of resistance to the antibody may be. So to summarize this complex picture of interactions, I'd like to share with you this synthetic scheme. So in this work, we observed how the binding of daratumumab to myeloma cells produces an increase in the transcription of genes related to metabolism and homeostasis, and on the other hand, a down-modulation of genes involved in the cell cycle and in the response to DNA damage. Furthermore, we also observed the release of microvesicles covered with antigen-antibody complexes and molecules involved in the production of adenosine, immune checkpoints, and complement inhibitory receptors. The microRNA content of these microvesicles is different from the microvesicles produced in the absence of the antibody, and their internalization in NK cells induces a reduction in proliferation and an increase in activation. This was observed both with functional and molecular experiments. In myeloid-derived suppressor cells, on the other hand, daratumumab seems to paradoxically produce and increase in the suppressive activity. However, this observation needs further investigation. So in conclusion, the take-home message is that daratumumab can act both in soluble form as well as bound to microvesicles derived from myeloma cells, interacting with cell populations of the immune system. Moreover, daratumumab acts both through direct binding to target molecule on myeloma cells and through FcR engagement on immune cells. These secondary interactions, in particular with NK cells and myeloid-derived suppressor cells, may help to explain the efficacy of daratumumab, but also the onset of resistance. And it is therefore possible to exploit this observation to devise new strategies and therapeutic combinations that can delay or prevent the ineffectiveness of therapy, thus improving patient outcome and survival. I would like to thank Professor Malavasi and all my colleagues that took part in this project, and of course, all of you for your time.
Fantastic talk, Dr. Angelo. It was really someone new to the field, especially for in the field of multiple melanoma. I think I learned a lot. So to begin with, everybody, like whoever would have any questions, feel free to jump into the chat box and ask your questions. And just to begin with, I would like to ask a very basic question. Given like the field of MM, what do you think would be the future of immunotherapy? And you mentioned that there are some drugs, especially the antibody, the monoclonal antibody that you motivated in the beginning is currently in the clinics right now. I was just curious, like where does the field with respect to immunotherapy right now it is standing, and what are like the routine therapeutics or chemotherapy that are being administered currently to the patients who come into the clinic with multiple melanoma?
Thank you for the questions. I don't know if I am the right person to answer these very specific clinical requests, because I'm not an hematologist, a clinician hematologist, and my work has always been more in the lab than in the clinics. But of course, the therapeutic antibodies were a great improvement in the therapies of multiple myeloma, since, as I told you, you always have to change the compound you use, because eventually the disease becomes resistant. So developing therapeutic antibodies which can be specific to a target on myeloma cells is a great news, something important also, because you have to consider that the chemical compounds may have also a lot of side effects that a sort of natural molecule like an antibody does not have. So of course there can be allergic reactions and everything, but the help that therapeutic antibodies have brought to the field of clinics, I think, is huge. Maybe Dr. Cerchione can be more specific to answer your question, but
no, yeah, that does open up a little bit. Yeah, definitely. Thank you so much.
Immunotherapy is the present, not the future of multiple myeloma. I think that currently we are better knowing the biology of our patients, and this was the start to better counteract the complications of multiple myeloma, which unfortunately today is a not curable disease, but we can manage it in a wonderful way. And the key of the success are not only the treatments, but the combinations of the treatment, because we know that myeloma is heterogeneous, is a really heterogeneous disease. As you can see also from the programs of the event we planned together, we can consider as multiple myelomas. Myelomas are different in way of presentation, in symptoms and signs, and also in response to the different drugs we have available. That's why maybe starting from the mechanisms of drug resistance, we can discover how to better attack all multiple myelomas. And if compared with other oncologic and hematological disease, what we lack, and this is a key message for the researchers who are following us today, that in multiple myeloma we don't have a real target therapy. I think that we should better define some targets, as for example, BCMA or CD38, in order to better discover which is the best way to attack not only the myeloma cells, but the microenvironment. This is the key, and I think that we have achieved in last years wonderful results if compared with 10, 15 years ago, but we need to do something more, because now we have the drugs. Drugs are effective, but we should know which is the exact patient for that drug. And maybe we should go in another world of multiple myeloma, that of personalized medicine. I think this is the near future of this disease. And I think that we could work together also, starting from really interesting data as the ones from Angelo, discovering better the biology and the mechanism of drug resistance in order to improve the management of our patients.
Exactly, like I totally agree with that. And I think we have a question in the chat box.
Yeah, I think I can see it.
Okay, awesome.
Did you come across small molecule blockers of microvesicles formation in multiple myeloma? Actually, yes, we did some experiments which are still ongoing with Dinosaur, which is a blocker of MV formation, as I'm sure you all know. But still, we don't have solid results to show, actually.
Awesome. Sarah has another question, and she's asking, I might have missed this, but did you look at HSPGs in melanoma?
Excuse me?
Sarah is asking, did you look at
Okay, yes, I can see it now. No, actually, we did not.
Okay.
But thanks for the suggestion. We might be investigating with that. Thank you very much.
Yeah, it's so interesting that after giving the drug, you saw an upregulation in the secretion of the microvesicle formation, right?
Yes, that was nice. And there was not change in the size of the MV, so I think that they all come from the membrane of the cells. But there was big change, a dramatic change in their content, so in the microRNA content, and also in their concentration. So I think that Tumumab enhances a lot the detachment from the membrane of the lipid rafts that contain not only CD38, but also other molecules, as I show, CD39, CD73, but also many other molecules belonging to different families. And I think this is another mechanisms of action that we should add to the known ones for what daratumumab is concerned.
Exactly. And like given the field right now with the EV being more like at a niche stage and then using that in conjunction with immunotherapy, I think it's just gonna boom up and there is a lot of possibility in future.
Yes, I hope so. I hope that these will become something that's also useful in clinics, which is not just the results, the good result in the lab, but is also something that can be used to improve patients' health. I hope that for the future.
Exactly. And given that if we start like looking into the GMP facilities for EV production and maybe even utilize EVs as therapeutic carriers for the drug delivery in the multiple myeloma patients could also be one avenue to look into.
Yes, of course.
Awesome. Yeah, so I think we are good on time and we addressed all the questions from the chat box as well as everybody who had questions. And Yeah, if anybody also has any questions regarding SOHOItaly or SNEV, feel free to ask us. And, Claudio is here, so anybody can ask any questions on how to be a member or anything. So feel free to ask about that too.
I tell them how can be a member. You can go on www.sohoitaly.it and you can go there. The subscription is for free. We don't want absolutely to take money from young investigators, and we use the funding from pharma companies, from society, to promote our activities, and we are really proud to offer everything for free, educational of high level. I think that is something that we really need in a world in which the big conferences have high cost, and every the educational programs have also high cost. So I think that SOHOItaly could be a new reality. Moreover, Angelo is the proof that we really want to promote the young investigator works, and this is another thing that I want to underline about our societies. This is built together with the young researchers and these pro-young researchers. Angelo not only had the possibility to present today to your great organization his data, but will have also the possibility to be guest of MD Anderson Cancer Center. Thanks to the contribution of SO, he will go in next SO of USA, so in Houston in next September, to participate to their conference for free. They will give to Angelo accommodation, travel, and so on. And for every event, we want to promote a research of one young investigator. We have this voting for every event from the abstract pool that we start in all our conferences and workshop. And thanks to SO USA, we can offer a great scientific prize. We don't want to give prizes in money, but we want to promote the scientific development of our researchers. And I think that going to Houston for a hematology researcher is something that everyone have dreamed in their life. So I think that you can start to subscribe to our activities. We are developing also a new website in which you can find also the educational activities of our last conferences. Moreover, we are in connection with the biggest channels in medical education of United States, such as VUMEDI, VUJ, and Monk. We are their partners, so you can find also about multiple myeloma, last presentation, also of extracellular vesicle event on these channels and also on our website. Moreover, we are in social networks, in Facebook, Instagram, and LinkedIn with our page. So if you have curiosity after having seen our activities, our presentation, I'm available. The other member board are available to clarify your doubts and also to wait for your suggestion or your proposes. We are totally open. SOHOItaly is not our creator, is of everyone. So we can develop together something of interesting. And I hope that we can also have other events in partnership, SNEV and SOHOItaly, because I really like and appreciate the freedom and the freshness of your society and of your board members. So it's a pleasure to collaborate with you, and it was a pleasure this workshop today.
Absolutely. It was an honor to have you guys here, especially like this pandemic let us happen this, right? Like we got more connected and then things became more connected in this virtual world. And we got this big exposure of so many societies, which we didn't even realize existed a year ago. So it's really an honor for us at SNEV, from every executive member in our committee, to host you guys and have this workshop today with us.
Thank you.
Thank you very much. Yeah, thank you very much. And
Thank you
to everybody who joined in today's call. We'll be having this video uploaded both on our SOHOItaly's YouTube channels as well as on our SNEV channels. So feel free to like it, share it, and comment on it.
Yes. I want to thank all the people who were connected, and I hope that we will see soon with other activities. And I want to thank in particular Kshipra for her wonderful organization work.
Thank you so much, Claudio. It was an honor. And it's, as I said, it's not just one person. We are like a big team behind to like make this event happen. So yeah, really
Yes,
appreciate.
Also from my side, thank you very much for the organization, and to all of you for the time you dedicated to listening to me. And I hope that there will be other chances to get back together.
Definitely. I think we should definitely have more collaborative events between SNEV and SOHOItaly to like keep it going.
Thank you.
Cool. Awesome. Thank you very much, guys. Bye.
Thank you.
Bye. See you soon.
Bye. See you soon.